Hello, everyone. Welcome to today's live broadcast, the FDA's experience with emerging genomic technologies, past, present, and future. I'm Julianne Chiat of LabRoots, and I'll be your moderator for today's event. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them into the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing the presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button on the lower left. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. I would now like to introduce today's speaker, Weida Tong. Dr. Tong is the Director of the Division of Bioinformatics and Biostatistics at the FDA National Center for Toxicological Research. This division of the FDA develops bioinformatic methodologies and standards to support FDA research and regulation and to advance regulatory science and personalized medicine. Dr. Tong has served as a science advisory board member for several large projects involving multiple institutes in Europe and the U.S. He also holds several adjunct positions in universities in U.S. and China. One of the most visible projects from his group was leading the MicroArray Quality Control Consortium to develop standard analysis protocols and quality control metrics for emerging technologies to support regulatory science and precision medicine. Other projects include the development of a molecular toxicity knowledge base for drug safety and the development of the FDA Bioinformatics system, ArrayTrack TMC, to support FDA review and research on Pharmacogenomics. Dr. Tong has published more than 200 papers and book chapters. I will now turn it over to Weida Tong for his presentation.
Okay, thanks, Julianne, for the kind introduction. And I also like to thank Dr. John Qualkenbush to ask me to talk in this virtual conference. And honestly, I have never done any presentation in any virtual conference. And my webcam is broken, and so that really does not help. So this is going to be an experience. As Julianne mentioned, I work at the National Center for Toxicological Research, and this is a research center in the FDA. And in the past decades, I have had a privilege to work with some of the brilliant scientists and the reviewers in the FDA to deal with and the genomics data. So I'm very glad to have this opportunity to share some of our lessons learned from this process and with you. So what I like to do, I'm going to start to talk a little bit about the regulatory science, because this is the one of the white papers released by the FDA. It's a layout of some of the guiding principles for the regulatory science research in the FDA. Now, regulatory science is just one of the scientific disciplines, means different things to the different peoples. But in the FDA's mind, the regulatory science is the science of developing new tools, standards, and approaches to assess the safety, efficacy, quality, and performance of FDA regulated products. And now you can see this is a pretty mouthful and the definitions. Our former commissioner, the Dr. Hamburg, actually she provided a much more concise, elegant, and abstracted way to describe the regulatory science. This is what she said. I quote in here is regulatory science is the science to bring 21st century approaches to the 21st century products and the problems. So the question is, what is the 21st century approaches? So in my mind, 21st century approaches have two sides of the stories. And one side of the story is about the technology themselves. So to this audience, I don't need to remind you about the tremendous progress we see in the past decades related to the innovation in the biomedical and the science areas. Nowadays, and everybody talking about the next generation sequencing, and I'm not going to show you the graph shows how the cost of the sequencing was decreased, deviate from the Moore's laws, and we know next generation sequencing become much more affordable, and that this technology has been used now very routinely to interrogate the genetic the variants and to understand how these variations are related to the disease and the health. In the transcriptomics area, and we are no longer talk about messenger RNA. I think we still talk about a messenger RNA, but on top of that, we're starting to talk about the microRNA. We talk about long non-coding RNA. We talk about a circular RNA and other RNA species. And the reason we will be able to get into expand our horizon in terms of the study of the transcriptomics is because the technology and everything's really powered by the new technology and we have experienced with. And compared to the transcriptomics and the proteomics and the metabolomics, and it's still lagging behind, but a tremendous progress has been made in the past few years, and we're going to see a lot of the good things came out from these fields in the next few years as well. Now, beyond the omics, even some of the traditional technology we have been using for many, many years, and we're also starting to see the innovation, like a cell-based assay. Nowadays, we use routinely on the high-content screen assay, and this type of assay allows us to measure multiple endpoints and the different type of the cellular response to the treatment, and in the same times, and it's very, very useful. Of course, we also see the 3D cell models and organ on the chip, and so on and so forth. So, in my opinion, and we are not lack of the new technology. Actually, we have so many new technology. But once we have a new technology introduced to the researcher community, and everybody was rushing and try to use this new technology to address some of the issues difficult to be addressed using the conventional methodologies. And of course, the incentive is you might get a good publications by using new technology compared to the conventional methodologies. And in this publish and perish world, this is also very important. But very clearly, and during this process, and we're starting to realize some of the technology just not live up to the expectation as they are promised at the very beginning with. So during this process, some of the technology goes away, and others prevailed and become the standards. We know how to use this technology in the right context, and using the FDA words is a fit for purpose application. However, the concern we really have is the time to take the innovation to the regulatory application is normally very, very long, and usually we're talking about over 20 years. And by the time the technology becomes mature, the new technology coming in. So another side of the story for the 21st century approach is how the government agency can play a role to expedite the transformation from the innovation to the regulatory application. And clearly, we cannot do that alone, and we need to engage to the research community and stakeholders to optimize the reproducibility of the technology, and to standardize the analysis protocol, but most importantly, and to encourage the data transparency. So actually, FDA has been doing that for a long time. And 10 years ago, when the microarray is the only game in town, and with respect to the high throughput genomics technology, and the FDA already starting to dealing with the microarray data. By that time, many pharmaceutical company and use the microarray technology to assess the drug safety and to look at the biomarkers from the clinical trial. And they came talk to the FDA, says, hey, we generate a lot of the genomics data. How you view these genomics data in the regulatory setting, how our interpretation can be used to support the drug submissions. And by then, an FDA really did not have too much experience with the microarray technology. And we did manage it to release a guidance document called Guidance for Industry on Pharmacogenomics Data Submission. And within that document, we defined a novel data submission program called the Voluntary Genomics Data Submission, or the VGDS. Later on, we're starting to see the data beyond the microarray. We see the data from the proteomics, from the metabolomics, and also the data is not belong to any omics. So we changed the name called the Voluntary Exploratory Data Submission, or the VXDS. Just to avoid the confusion, I'm just using the VGDS in these presentations. So, and using VGDS, and the sponsor will be able to approach to the FDA out scope of the regulatory application, and we will be able to discuss, to define the best way to interpret the data and to analyze the data from the microarray experiment and beyond. So, and I forgot which slides now. So, okay, so that works. So, and we set up the 3 different objectives in the VGDS program. And first, of course, we need to have a data repository to capture all the information and all the data submitted by the pharmaceutical company, and because we believe these kind of the data are important to support our future regulatory policy. Second, we also wanted to reproduce the sponsors' results to understand how the sponsors to reach their conclusion based on the data that came out from machine. And lastly, and we also wanted to do our own analysis to present the agency's view on how this kind of the data should be analyzed, how this data and should be interpreted. So, and as a research arm to the regulatory centers in FDA and NCTR as an integral part of the VGDS program, and my group has really taken on all these 3 objectives and the list on these slides. So, for example, and we develop a software environment called ArrayTrack. This is an integrated solution for the microarray data management, analysis, and interpretation. So this tool, we also made this tool publicly available. It's widely used by research community, and still till today. Now we are in the process do the facelift, because now we try to make these tools can be used for the RNA sequencing data. But anyhow, and we did a lot of the trainings, and a lot of the FDA reviewers use these particular tools to conduct analysis of the genomics data. And as I said, my group also did a lot of the analysis, particularly, and we did independent analysis. We often found a slight change of the statistical method we applied, and the results is not even close to the results compared to the results submitted by the sponsors. But the most troublesome part for us is often the different results and the lead to very different biological interpretations. So, and this particular issues is not just exist in the communication between the pharmaceutical company and FDA. If you look at the literature, and actually these issues have been brought up in the many publications. And this slide just summarizes the 3 key issues we try to address in our review process. The first is the quality control issues. And every time we receive data from the pharmaceutical company, and the data is reasonably quality controlled, the question is, how good is good enough. And so we really don't. We need to set up some sort of the baseline so the company will be able to follow and how to conduct the quality control. The data analysis issues, this is much more serious than we originally anticipated because, as I said, the slightly change of the methodology will lead to very, very different results. And even today, and for the data we see and from the next generation sequencing, it's very much the same story. The question I listed in this on this slide, even though we did that 10 years ago, but equally applicable to the issues we are facing for the next generation sequencing or other omics technology. So the question in terms of the data analysis, we are trying to provide a baseline analysis method to inform the company. It says, okay, you can do the analysis using the methodology, whatever you choose, but you need including the analysis using that particular methodology, so we will be able to compare your results and with others. And lastly is about the cross-platform issues, and this is always, we cannot avoid that. If anything can make money, you always find different version of the same stuff, right? For the microarray, at least at the very beginning, we have so many different platforms. So the question is, if I give the same samples to the different platform, are these platforms going to give me the same biology or not? So those are the issues we try to address, and we certainly realize this cannot be done and within the FDA. And we really need to talk to the research community. We really need to talk to the stakeholders. So that's why we established MicroArray Quality Control and a consortium, or called the MAQC consortia. And this is the FDA-led community-wide consortia. And we have all the FDA centers and participated in this as a consortia. And we also have a lot of the participations from the different stakeholders, the research communities, and the government agency, and so on and so forth. And the objective of this consortia is to assess the technical performance and the practical utility of the emerging technology, particularly in the genomics technology, including the microarray and the next generation sequencing. I'm going to talk a little bit more about the next generation sequencing later on. And we are specifically on the interest on how this technology is being used in the clinical application and for the safety assessment. And we try to make as transparent as possible because that this is a government-funded project, and the transparency is extremely important. Of course, we do something routine, and all the results and the conclusions are going to be published in the peer-reviewed journal. So this is basically everybody is doing, and we are doing that as well. And at this point, we published a little over 30 papers, and 1/3 of them is published in the Nature Biotechnology. And every time we have a publications, we make sure that data is freely available to the public. And whatever possible, we also wanted to make sure that biological samples we use to generate this data also and publicly and available. So, and we started the first consortia project in the 2005. And we conducted the first phase of the study for 17 months. And we have 137 participants from the 51 organizations. And in this project, we look at the issues related to the microarray, and more specifically, we try to understand whether microarray technology is reliable or not. So, and we did a cross-platform and the comparison, and we compared the microarray results with 3 alternative quantitative gene expression platforms, including the real-time PCR, and we looked at the choice of the bioinformatics methodology, how they can impact the downstream biology, and we looked at the quality control issues, and all of them is really focused on one word, and the reproducibility. We wanted to understand how to generate the reproducible results using the different microarray platforms, using different bioinformatics approach. So in the end of the day, and we have 6 papers and published in the Nature Biotechnology, and some of the key findings are used to develop the companion guidance in FDA for the industry on pharmacogenomics and data submission. So nowadays, and you probably heard a lot about a lot of the discussions about reproducible science. For example, some reports indicate up to 85% of the resource are wasted in the irreproducible science. And some study also indicated irreproducible results we see in the high impact journal just as many as we see in the lower impact journal. In 2013, actually Nature put together a special issue to discuss about the irreproducible science. That's on the right side of the slides. There is a lot of the topics being discussed, and they pointed out that irreproducible biology research really costs about $28 billion per year. The journals call a unification to develop a strategy to address the reproducibility issues. NIH even step in to put a plan forward to make sure that NIH and funded project that the results came out from this project can be verified by third parties. And you know just as much as I know, when we do a scientific research, if we try to reproduce other people's the results, and we often find that the resistance in publication by the journals and by the reviewers, they might come back and say, so why you do that? And this work already been done by someone else, and that your paper is a lack of innovation. So one of the paper included in this Nature special issues and talk about if a job is worth doing, it is worth doing twice. I think this is very true for the MAQC project, because in the MAQC we are not focused on innovation. We are more focused on developing study design comprehensive and systematic enough to address some controversial issues. So if I can highlight in the one contribution of this project or one lesson learned from MAQC-I to the research community is about the reproducibility. And the conclusion we draw from our study, and we finally realize, and the reproducibility is a third dimension beyond the sensitivity and the specificity we normally used to assess the accuracy of an experiment. And the reproducibility is not a common language usually used by the statistician when they try to design an experiment and so on and so forth. And for the microarray data, and we found using the very simple volcano plot, that means the fold change plus the p-value, and we will be able to balance the reproducibility and the sensitivity and the specificity, even though there is still debate what particular cutoff we're supposed to use for the fold change and for the p-value, but a combination of these two values does provide or does enhance the reproducible results from the microarray technology. I highly suspect, and we have not been tested yet, and that these combination criteria and also can be used for other omics technology as well. So after finish MAQC-I, and we move forward to the second phase of the MAQC project. And in that project, we're starting to look at the clinical utility of the microarray technology. And more specifically, and we try to understand how the gene expression-based classifier or predictive models and can be used in the clinical setting. Now, this is a particular and timely important when we started this project, because by that time, the MAMA print and for the breast cancer, and submitted to the FDA for approval. And we are trying to gain a little bit more or better understanding how these gene expression-based biomarkers and can be used in the right context for the clinical applications. So we started in September 2006. We have a little bit over 200 participants from the 86 organizations, and we published 13 papers, and some to the Nature Biotechnology, most to the Pharmacogenomics journals. On the right side, you see the box plot, and on top of the box plot, you see the numbers. Those are the 13 endpoints, and we look at it in this project. And the y-axis is prediction performance. And in the middle, from the K to F, is the clinical endpoints for the breast cancer, multiple myeloma, and the neuroblastoma. So I'm not going to go in great detail to explain the results. And I have a 4 bullet point in the bottom of the graph and really summarized the key findings from these studies. And first of all, we found that the prediction performance is really endpoint dependent. If we work with the easy endpoint, and all the prediction is good, and regardless which methodology you use. If we work with the difficult endpoints, that's for example, random numbers. None of the methodology is going to work. However, if endpoints and it's between the easy and the difficult, in the between, sometimes some methodology might show a little bit better. But overall speaking, the choice of the machine learning method does not play a major role in terms of developing the statistically significant classifiers. Okay. And the second bullet point is we also found that the multiple models can reach the comparable performance by using different gene set. And there is it's very difficult to separate which model is better than another one. And in the machine learning world, we always doing the training, using the training set and do the internal and the validation. So the question was asked, so whether the internal validation results have any relevance to the external validation performance. That means internal validation using the training set can be extrapolated to estimate the performance when the model see the sample and never be included in the training set. And on the box plot, you can see the yellow is from the internal validation, and the blue is from the external validation. So I think they looked at 13 endpoints, even though there is a surprise here and there, but overall speaking, they are very consistent. Okay, and we also look at the simple machine learning method and compare with some complicated using the nonlinear methodology and so on and so forth. And basically, our general impression is a simple data analysis method just perform as good as the complicated methodologies. So when we working on the MAQC-II on the next generation sequencing, the 2008 slowly take the center stage in the biological research, and particularly there is a paper published in 2008 in the Nature Review Genetics and talk about the RNA sequencing, and the review paper listed the several advantages of RNA sequencing over the microarray, and the paper forecasted that RNA sequencing is going to replace the microarray in a very short period of time. But at the same time, the Nature Biotechnology also released an editorial to call a consortium efforts, because what the Nature Biotechnology see about the next generation sequencing in the 2008 is very much the same situation we see in the 2005 for the microarray, and we should have a way to benchmark different technology and to compare, particularly focus on the reproducibility. So, and for that, we started we started the third phase of a MAQC project, and also called the Sequencing Quality Control, or the SEQC. And we just completed this project last year, and we have 180 participants from the 73 organizations. We generated a little bit over 10 terabyte data. By the time we deposit the data to the GEO, we notice, and our data actually occupies 6% of all the RNA sequencing data in the GEO. So we're very happy because we feel, and through the many years study, and we contributed to the researcher community, and by sharing the larger volume of the data with the researcher community. And we have 10 paper published, and 3 by Nature Biotechnology, 2 by Nature Communications, 3 by Scientific Data, and 2 by Genobiology. Because the time, I'm not going to go through all the results because there was a lot to talk about it. So what I'm going to do in the next 5 slides, I'm going to list top 5 findings, and just like a TV show. So number 5. So in this study, and we have 6 reference samples, and it shows on the left side is A, B, C, D, E, F, and we distributed these 6 reference samples to the 11 laboratories. And some laboratories have all the 3 machines, Illumina Solid 454. Some just have 1, some have 2. So whatever the machine they have, they did all of it. They did all of these 6 samples. So using this mechanism, we will be able to see how the results are compatible across laboratory and across platforms. On the right side, I'm just show you some of the interesting findings. Very, very simple analysis. Actually, we did a much more complicated analysis that's published in the Nature Biotechnology. And you can see if we look at the sample A on the same samples, and was done the sequencing in the 2 different laboratories, and then we took the absolute value from the laboratory 1 compared to the laboratory value 2, and we found, if we consider it's a differentially expressed gene, compared same sample from different laboratories, we found so many genes can be considered as differentially expressed, okay, up to 10,000 of them, and this is not affected by the choice of the bioinformatics method, and the results are always the same. However, if we look at the ratio data, and from the lab 1 compare lab 2, the ratio data here is A versus B, and the results are much more consistent. So we feel the relative measures agree well across the laboratories and across platforms, but not for the absolute measurement. And we also did a very extensive comparison between the RNA sequencing and the microarray. And clearly there is 2 groups in our consortia, and some is really like a microarray, some is really like RNA-seq, and we have a constant debate. So we designed several experiments. This is just one of them. And in the middle, you see, it's on the graph, you can see the 3-letter squares, on those are the compounds. And we select the 15 compounds. And for each compounds, we treat 3 animals and compare to the matched control. So we will be able to determine which genes can be considered differentially expressed for each individual chemicals. And if you look at it closely, for every 3 chemicals, and we deliberately choose these 3 chemicals and falling into the specific mechanism that lead to the toxicity. So using that way, we can compare between these 3 compounds, and also compare the one mechanism to another mechanism. We did all the microarray. We also did RNA sequencing. And the results are a little bit more complicated. Let me just try to get it quickly explained what the results looks like. So if you look at the graph A, that's at top left, the graph A, so the y-axis is the concordance. It's just basically it's a Venn diagram between the differentially expressed genes identified by RNA-seq versus microarray. So this is just a simple Venn diagram, shows on the y-axis. On the x-axis is a number of the gene differentially expressed using the RNA-seq methodology. Now you can see some compounds only elicit 100 genes differentially expressed. Some compounds elicit a larger number of genes differentially expressed. So from the least number to the most number, about 10 folders. And we found that the linear correlation between the concordance between the microarray and versus the number of the genes being considered differentially expressed. If you look at all these 15 compounds, this is basically the graph A, and shows in here. So this makes us wonder what's really going on. Why? It depends on how the biological systems are badly screwed up, right? If you badly screw up, now the concordance is higher. So what we did next is pretty interesting, and we look at the gene expression intensity or the gene expression level for all the genes, and then we split those genes to two parts. And using the median, and above the median we consider it's highly expressed, and below the median we consider lowly expressed. Now this is the result shows in the graph C and the D. Again, the y-axis is the concordance between the microarray and the RNA-seq. You can see if you look at the above median and the expression genes, the concordance is very, very higher, regardless of how many genes are differentially expressed for each compound. So regardless of which compounds we look at it. If you look at the graph D, the concordance is much lower between two platforms if we only look at the lowly expressed genes. So very clearly, through this analysis, we know the major difference between the microarray and RNA-seq is the sensitivity to determine or to accurately determine the lowly expressed genes. So we did a real-time PCR, which has to confirm what the results looks like. It is a graph B. And without going into any detail, our conclusion is RNA-seq has a better sensitivity for weakly expressed genes than microarray. So, and number 3, so when we use the RNA sequencing, and one of the incentives is for the gene discovery. You know, and for the microarray, what do you put on the array? What do you see? What do you got? But for the RNA-seq, the supplies are all over the place, and you might find some of the new genes. The question is how many reads we need to identify new genes or the novel genes. So the bottom two graphs really show these results. Now on the left side, we only look at the known genes, and we increase the read depth from the 10 million to the 10 billion. And then you can see for this graph, the y-axis is the number of the genes being detected, and you can see, and as the read depth increased, and the new genes being continually identified. This is true for the known gene as well as for the junction that represent the new genes on the right side. And every time when I give a presentations, and I always being asked to say, oh, which particular pipeline and should I use? This is another effort in our project, and we looked at 13 different software package, and we twisted different parameters. We came up with 278 and the permutations. And those permutations relate to the mapping, quantification, normalizations. And we looked at all of these combinations, and we compared the differentially expressed gene versus real-time PCR result. We also looked at classify. We drew a number of the conclusions. And looks like I'm starting to run short of the time. Let me just go a little bit more faster. So what we found is if a pipeline did a good job to determine the differentially expressed genes, usually that pipeline will be equally effective for classifier development. So number 1, and this is a pretty interesting investigation, and this particular idea really come from when I went to the meeting, and I asked a speaker, says, you know, on RNA sequencing was used and pretty common now, are you going to, using the RNA sequencing, go back to sequence all the old samples? And the answer is very obvious, is no. So really boiling down is how we're going to dealing with the microarray data in the RNA sequencing era. Okay, so there is two angles to look at these issues. And first of all, we have a lot of the biomarkers being developed in the microarray area. And for example, the MAMA-print, as I mentioned earlier, we did a lot of the clinical trial has been done. These genes, these biomarkers have been extensively used in the clinical practice. But if we in the future and everyone do the RNA-seq, not doing the microarray anymore, and how are we going to do this MAMA-print biomarkers? It can be used in the RNA sequencing data or not? So this is the first question. Now, let's just envision in 10 years down the road, and everyone using RNA sequencing data, and a lot of data being generated, a lot of the biomarkers are available. So the question then is how we can go back to the microarray data to leverage the past investment. So this is the papers and really address these particular issues, and we publish in the Genome Biology. And basically, we look at the signature genes. We also look at the models. And in terms of the signature genes, we can easily to apply the same signature genes from microarray to the RNA-seq and in reverse. For the models, from the microarray to the RNA-seq, it's much better. So you develop a biomarkers using the microarray data, and then you predict the sample identity based on the results from the RNA sequencing data. And that's actually performed very well, but the reverse is not true. So to this point, and I really talk in the very general terms, and the devil always in the detail. So I really encourage you to go to our publications and download the data, do your own analysis, draw your own conclusions. So where we go from here, I do want to point it out on the nowadays, everybody is talking about the next generation sequencing. And in the FDA, we also have a next generation sequencing and a working group. And the next generation sequencing is being used in a variety of ways. So but to my opinion, a next generation sequencing is just a tool, right? And when we're talking about the challenges and the issues and the regulatory applications, really based on how this tool is being used. If you're dealing with the human genome, that's we're talking about the 3.2 billion base pair, and the challenges, the issues will be entirely different when you're dealing with the microRNA. That's only 18 to 25 base pair long. So in the SEQC project, and we did quite a broad survey and tried to see how the next generation sequencing tool was being used in the research community. We found that 50% is in the DNA sequencing, and the 25% is RNA sequencing. And we see a little for the microRNA sequencing, and clearly microbiome has been used tremendously on this particular technology. And in FDA, we also use the next generation sequencing for the pathogen identification and outbreak detection. But the portion still is small. So what we decided to do is, and we said as a natural extension of the SEQC1, and we decided to, in this new the project, and we call the sequencing quality control phase 2, and we're going to focus on the whole genome sequencing and the targeted gene sequencing. And we're doing the same common things. We look at the quality control matrix, we look at the reproducibility, and we also look at the bioinformatics. And I'm not going to go to the milestones, and I just want to point it out. And we will have our first public 2-day workshop to discuss this project in an NIH campus in September 13 and 14. And everybody is welcome. And this is our first and the workshop, and we try to form the consortia in that workshop, and also we try to collect that input to finalize our study design. I hope I can see some of you and in this meeting. So just want to point it out, and when we decided to do this project, and we have struggled quite a bit because for the whole genome sequencing as well as the targeted gene sequencing, we started with the samples and all the way to the clinical application, the decision making, and that involved many different steps. And from the sample to the DNA sample, that's really involved the lab efficiency. From the DNA to the data, that's involved, and that's really being determined what particular next generation sequencing platform you're going to use, and short reads versus long reads. And from the data to the interpretation, for example, single nucleotide and variations, that's, CNVs and translocation, that's really also up to what particular bioinformatics methodology we use. So there's a lot of the issues we try to sort it out. But in the end of the day, and we put together a draft study design that involves 5 components that shows on the right side, and this is the one we're going to present to the consortia in the September meeting at the NIH. And the first one is more focused on the germline mutation, and we look at the SNV, CNV, and the indels. And we're also going to look at the short reads versus long reads, and how we can combine short reads with the long reads to achieve a better results. And the third project, we're going to look at the detection power for the rare mutation using the targeted gene sequencing. And we're also going to pay specific attention on some of the specific genes, because we know the next generation sequencing is not a technology can solve all the problem. And for some genes, it is very, very difficult for the NGS technology. For example, like HLA genes, the P450 genes, and some of the genomics region, like a repeated region, GC-rich region, even short indel, and sometimes the NGS cannot get it entirely right. So we wanted to get a sense how good the NGS technology can achieve at its current levels. And lastly, we're also going to look at the MAQC-I, and that particular machine was approved by FDA as a biomarker machine for support of precision medicines, and we see where we are in terms of that machine. So I just going to skip those 2 slides. Those are the data sets we're going to use, and please feel free to send me the email, and I can explain to you why we choose these 2 data sets. I'm just going to go to my final slides. I want to point it out, and the bioinformatics, it is really important piece of the entire puzzle. And when we go through from the microarray to the next generation sequencing, and we often found that the results are very, very difficult to be reproducible. And sometimes we only can reproduce others' results by acquiring the original script. So, and also we found that in silico mistakes are quite common, and because nowadays we are no longer look at the data closely. We never look at the Excel spreadsheet anymore, and we just apply the algorithm on an Excel spreadsheet. And sometimes Excel spreadsheet turned the number to the date, and we didn't even notice, and we applied the algorithm, and we thought we get good results. So we do need to have some sort of the best practice, and we need to implement the positive control and the negative control to interrogate the algorithm we use. And we see this really, really common in our consortium efforts. And certainly the general rules can be followed, and the best practice can be developed in the bioinformatics fields. And I also want to point out the NGS is just a tool. And so we should avoid to talk about it in general. And when we talk about the challenge and issues, we really need to talk about how these tools are being used. And also, and I feel, particularly, and when we work in an FDA, we really feel that we need to talk to the researcher community. We really need to talk to the stakeholders to gain consensus to produce the reproducible the studies. And with that, I like thanks and all the consortium members and contributed to this project, and some of them, I just pull out one slide that this is not to try to be inclusive. And some of you probably did not see your logo on this slide. I apologize here. And finally, thank you very much.
Hey, thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them into the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Our first question. Wondering if the sensitivity of RNA sequencing is contributing to normalization of the number of cells. If the microarrays are normalized for the cell number, does this change sensitivity?
This is a fantastic question, and I have to say, no, we did not look at that particular analysis. I know in Juncano and from Japan, they developed a per cell method and to dealing with these issues. But in here, we have not. But in terms of the sensitivity or the difference in sensitivity between the microarray and RNA-seq, I think that the conclusion we draw probably very much the same, because in here we really is apple versus apples, and both microarray and RNA-seq does not count the cells, number of the cells.
The next question. As differential splice has some time before finish, when do you think NGS and microarray data shall be finished?
Sorry, which question you are? Say again. Which question you're on? Is it number 2?
It is number 3.
Oh, number 3. Oh, sorry. As differential splice have some before finish, when do you think NGS? I'm not sure I understand the question, though.
That's all right. We can move on to the next question. The next question, number 4.
Okay.
All the data presented here appears to be on a population of cells. Do you have any guidance for single-cell analysis?
No, I certainly not. And this is a good question. And it's correct. We're using the pool of the cells, and we have not done anything on the single cells. This is another area, and it's really growing fast. Tremendous progress has been made, but we do not have any experience with it.
Okay. We have one more question. Has the FDA consortium meeting in September 2016 been announced somewhere?
Yes. Good question. And we are going to have a Federal Register announcement, and it's going to be announced very soon. And of course, and anyone is interested to participate, we already have the agenda and all of that. And you can send them an email. I will send you, and also I include in your email distribution. And in our consortium, we have a little bit over 1,000 people, so we have an email listing, and we're certainly going to distribute it to our existing email list. But if you feel you are not part of our email list, please feel free to send me the email.
Great. There are no more questions. I would like to once again thank Weida Tong for his presentation. Do you have any final comments?
Not really. Thank you very much. And as I said, this is quite an experience, and I've never done that before. And I hope and I delivered the message and clearly and concisely, and that the information is informative. And thank you very much. I do want to thank Don and Juliana and to set this up.
Okay, thank you. Today's webcast will be available for on-demand viewing through May 11, 2017. You will receive an email from Labbers alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Thank you and goodbye.